structure of monocotyledonous seed. So, monocotyledonous seed contains single cotyledon inside them, and this cotyledon is papery in its nature. And one more thing, this papery structure may be like a shield like organization that is called as scutellum. As this cotyledon is not storing the food materials, which is thin in its nature, and the food materials are present in the endosperm. That is the reason why majority of the monocot structure is occupied by endosperm. In the monocot seed, majority of the portion is occupied by endosperm. So, this endosperm is laden with food materials, and the embryo is present on one side, on the lateral side, occupying little space. So, if you observe the structure of the seed of monocots, so major portion is occupied by endosperm, and this is the embryo part. The embryo contains the scutellum, that is a cotyledon, single cotyledon is present. In this is a plumule which is having a cap-like structure called as a coleoptile covering like structure, and this is a radical part. So, which is enclosed by a sheet like structure called as coleorhiza, and here a specialized layer is present which is separating the embryo from the endosperm. So, this is called as alluron layer. So, this alluron layer is protein rich layer, and it is triploid in its nature. Endosperm is also triploid in nature, whereas embryo parts are diploid in nature. So, in monocotyledon seed, two different types of triploid structures are present and one type of diploid structure is present, whereas in the dicotyledonous seed only endosperm is triploid, whereas embryo is diploid. So, here alluron layer is a extra triploid structure that is present in the monocotyledonous seed. Actually, alluron layer is also being formed from the endosperm tissue only. That is the reason why it is triploid in condition.